And we've made it to the very last part of Bayesian analysis. For the last couple of moments, we're going to discuss what you do with the results that you get from a simple tree search in Bayesian tree space. All right, make sure that you understand the concepts from the last topic or the couple of topics, and we are now going to be putting it all together at the end. So previously we talked about these 5 taxa that we are interested in understanding in terms of their evolutionary relationships: the rodent, the frog or amphibian, a bird, a fish, and a human. And let's take a look at what this is going to give us. So all of you should remember that we talked about the hot and cold chains, all right? These are going to be the results of an MC³ analysis. First, the cold chain will give you all of the different trees that have been saved in memory. In fact, it is the cold chain through many, many generations that is surveying tree space and saving those trees in the computer's memory. The hot chains that were used to navigate tree space are discarded. Since they don't save trees, they don't need to be used again. And then finally, all the trees are saved in memory, which you can then use to interpret at the end. And I will make a final note that the program allows you to save every few generations instead of every single tree at every generation. And part of this has to do with the amount of memory that gets taken up by saving all these trees. Some of it has to do with the fact that many of the trees that you've sampled, especially at the beginning of the analysis, might not be very good. And so you are allowed to discard trees. You do not have to save every single tree from the analysis. And in fact, that applies to every other type of analytical tool that we've talked about as well: parsimony, maximum likelihood, and so on and so forth. All right, so here we've got a schematic, a diagram that you will often get when you're running these analyses. In this particular example, there are 200 different generations in which trees were searched. So 200 different individual steps through tree space. And you all will notice that this graph goes up and then reaches a plateau. Here and there, it kind of jumps up and down a little bit. It's not a perfectly straight line. The first 10% or so of most Bayesian searches results in trees that are not ideal or that have very low posterior probabilities. And part of the reason for this is that as you start to search through tree space, it's very rare that the program will start on those hills or tops where you will get trees with the highest posterior probability. Most likely the starting area in tree space is going to be in a valley somewhere. So a lot of the trees that are initially searched, a lot of those generations that are initially made or initiated are discarded, and we call these the burn-in. All right, so as soon as you've gotten to the point where most of your trees are pretty likely and have very high likelihood scores or probability scores, then you're good to go. And so there's no need to keep some of those poor trees. There are debates about this in the literature about how many you should keep and how many should be discarded. We will not be talking about the logistics of those. And then finally, when you get to the program that you're using, you're going to get some other outputs. So let's say that you've discarded your burn-in, those initial starting trees that were not ideal or that didn't have really high posterior probability scores. This is the output that you're going to get. Programs that we use typically are Mr. Bayes, very appropriately named, and there's another program that is commonly used called Beast. Mr. Bayes and Beast are the 2 main programs that are used for Bayesian analysis. They are able to handle these large quantities of sequence data, often large numbers of taxa, and handling all of the different chains running at the same time. In this output, what you all will notice is that there are 15 different tree options, 15 different topologies, numbered 1 through 15. And you will also notice on the right that there is a prior value given to each of those trees. If you refer back to the very beginning of this topic, we discussed priors. And I made note that you shouldn't really worry too much about these. The priors are any previous knowledge that you have about your taxa or about the relationships, which could include fossil data or any knowledge that you have about the rate of mutation in the genes that you are looking at. In this particular example, you will notice that the prior probability values are all the same for each of the 15 trees. Now, why are there 15 trees? These 15 trees are the 15 unrooted possible trees that you could get from these 5 taxa. And the prior probabilities are all the same. So before you go into the analysis, the prior was set by the program, or you could set it as each of these trees are equally likely. And the value 0.067 comes from the fact that each one out of 15 trees is equal to 0.067. All right, so each of the 15 trees has a 0.067 likelihood or prior probability. But after we run the analysis, is that really still true? So our prior beliefs or the prior assignments are not going to match necessarily what our posterior probability values are. All right, so this is the output that you will see for the prior probabilities, given all the different tree options and the selected model that was chosen. And finally, here's the output for both the prior and posterior probabilities. There are your posterior values on the right. You will notice that for the 15 trees, each one has a posterior value assigned to it. Some of them are 0. So that means that that particular topology, based on the analysis, has a posterior probability of 0. Based on our data, our sequence data, and the model, those particular topologies have a 0% probability of being the correct one. If you notice, tree number 1 has a really high posterior probability value of 0.964. And then tree number 4 has a value of 0.012, and tree number 13 has a value of 0.023. If you add those 3 values together, you will end up with a posterior probability value for all of your trees that equals 1. Remember that probabilities can never equal a value more than 1, right? So, for example, if you flip a coin, there's a 50-50 chance of flipping heads or tails. That's 0.5 and 0.5. You can never exceed 1. So, what this output is suggesting is that tree number 1 has the highest posterior probability value. It is probably the best tree and the most likely tree given our data. Those trees that are sampled or found much more in tree space have a higher posterior probability. And this makes sense because once you've made it to the hills and the top parts of tree space that we consider to have a higher posterior probability, it is very difficult to make a step down. So that suggests that just by chance, you are more likely to sample trees that have higher posterior probabilities, right? And that is the beauty of Bayesian analysis. The way that we search through tree space, we are basically forcing the program to look for those better trees. So the proportion of the time a tree is sampled is a good estimate of its posterior probability. So if we look at tree number 1, and we imagine that we saved or we sampled 1,000 trees in memory, 964 of those 1,000 trees looked like topology number 1. So it is the most likely. 964 of the 1,000 trees contain this particular topology. Now, how do we apply those posterior values to a clade? So here is the proper or correct topology for this particular data set, this is the topology that had the highest posterior probability, right? Where we've got fish, frog, bird, rodent, and human. And on this tree, I've assigned a posterior probability of a 1 to rodents and humans. And if you read the blue text, it says the posterior probability for a clade, remember this is the clade now, not the tree as a whole. The clade is the sum of the posterior probabilities of all the trees that contained that clade. So how does this end up being a 1? If we go back to the output of our posterior probabilities, let's pay attention to all of the trees in which rodent and human were present as a clade. So here we have those values again. And what you will notice, let's start with tree number 1, rodent and human form a clade. Same with tree number 4. And same with tree number 13. If you add up those posterior probability values, you end up with a 1. So if we go back to our tree, the rodent-human clade amongst all of the trees that were sampled, all of the possible tree topologies, that ends up being a 1. Rodent and humans have a support value or a Bayesian posterior probability value of a 1. I encourage you all to try to figure out what the posterior probabilities would be for each of the clades. So a challenge for you before we meet in person or face-to-face through our discussion sessions is see if you can figure out the posterior probability for the bird, rodent, human clade. And then try to go back and find the posterior probability for the frog, bird, rodent, human clade. If you can determine those values and you're able to read the output that looks like this, you will be in really good shape for any exam or quiz that tests you on this knowledge. So we are done with Bayesian analyses. I encourage you to ask lots of questions of yourself as you go through these slides again, and I look forward to talking to you all face-to-face very shortly. We are done. Good job for listening, and I'll see you soon.